So I know we're over time. And our next speaker graciously offered to not give her speech, but I'm not letting her off the hook because she has been one of the most helpful and inspiring people that I have met in my rare journey. And she's brilliant and she's a problem solver, and I can't wait for you to hear from her. So without further ado, Dr. Terry Jo Bichell.
Oh, thank you so much. I'll share my screen and I will try to make it snappy because I'm really interested to get to the discussion. And I really enjoyed these presentations. They're fantastic. In the agenda, the title of my presentation is "What do you want it to be?" And that's
Oh,
because I never gave it to Jill. But then I thought about that and I thought maybe that is a good title for the presentation
oh, that's
because yeah. So I am a neuroscientist. I became a neuroscientist because I have my youngest kid has Angelman syndrome. He's now 24 years old. And what we found in our quest to find treatments for Angelman syndrome was that biomarkers and outcome measures are a really crucial part of getting a drug from the bench to treatment in a person. And so we formed Combined Brain. Combined Brain is a consortium of 66 member organizations, and your foundation is a member, and we're very happy that you're part of us. And we work together to try to get whatever each of these different disorders needs to be able to get a treatment into the patients that they're trying to treat. So I always go over this slide, and this slide is basically a rough draft of what's needed to get a drug from diagnosis into a person. And so you obviously you have to have a gene ID these days to get a genetic treatment. But even for the repurposed drugs like Ethan talked about, it's really important to have the gene ID these days. You all have that, thank goodness. It's important to have your rodent and cell models. You heard from Jackson Labs and the fish model person. That's fantastic. You're already getting that together. You've got to collect natural history, and I'm going to talk about this a little bit more, but you've got to hear from the families to find out exactly what's going on across all the different families and all the different kids to be able to measure the right stuff in the rodents. Then you can start to figure out the mechanisms, and it's been really interesting to hear from the different labs today about how the mechanisms might be related to disease. Then you can get your idea for treatment that might come from repurposed drugs or gene therapy or a new drug or a new compound. But now you've still got more work to do. You've got to figure out exactly what it's like living with the disease. What's the disease burden? What exactly and how exactly will you do a clinical trial in kids with this disease? You've got to find biomarkers. For that, you need blood. Then you're going to have to work with animals, maybe mice, maybe non-human primates if you're talking about something like gene therapy or a novel technology. You've got to have patient-centered outcomes, meaning asking families something about what really matters to them to figure out if you've treated the right thing or not. And then finally, when you have all that, you can go and prepare your clinical trial and get it to the FDA. So this seems really daunting if you do it one at a time, but you're not going to do it one at a time. You're already doing it all at the same time. And that's the right way to do it. Just splattershot, go for everything. But what I'm going to talk about today is natural history. And as you can see, that is at the root of all of this. So in general, in the past, up until a few years ago, we in the neuroscience business were basically curing mice right and left. We did a fantastic job of curing mice, and we put the mice in the center of our research. And the problem then was that when we took the cures that worked in the mice, especially for neurodevelopmental disorders, and we tried to use those in clinical trials in human beings, the clinical trials failed. And the reason for that is because we weren't actually putting the human beings at the center of the whole drug development process. And what's really kind of cool these days is that the FDA also wants human beings at the center of the drug development process. They figured this out about 2017, and they've been publishing guidances since then, 4 really good ones about how you include patients in the center of drug development so our clinical trials don't fail. And when you do that, you ask the right questions to the animal models. When you do that, you come up with the right endpoints for the clinical trials, and then hopefully the trials will pass. So what I want to talk to you about is the power of natural history, the power of knowledge, and the power of data that all the families, all the patients have right now. And that is what you know about your kid, what you know about your family. It's a big deal. So there was a lot of power already in the fact that Cooper and Charlotte were in a family which right away put a lot of information about them and about FAM177A1 on the internet. That power that they used of knowledge about their own kids is already bringing in a whole lot more families. It's already pulling in information about what the gene does. I mean, already then they found the power of 6 patients when they uncovered the 4 siblings that were in the other paper that had already been produced in a couple years before that in this paper by, trying to move my picture here, Alazami. I was afraid I couldn't remember the name. So that was already up to 6 patients. And what I understand is that now you've already found 15, and that's probably old news. It's probably more than that already. And this is a lot of power. 15 patients already is enough. Like Ethan said, even 1 patient is enough to get to a treatment. Sometimes 15 patients is amazing. And the more patients you find, the more you learn about those patients, the more you're going to be able to find more, the more you're going to be able to treat more. So right now you already have the power to put natural history data to work, and you have it right on the website for the foundation. There's a link there for a movie that shows families how to do natural history on Rare X. And and you can, sorry, didn't mean to do that. That's what I get for talking too much, too fast. Trying to get through this as fast as possible. At any rate, the main point is you already can enter data into Rare X, and you already have that portal open, and you already have that information on your website. In fact, if you could put that link right in the chat, Jill or Jeff, that would be fantastic, so people can go right in and get into that. But what this will do is give you the power of more knowledge, because most of these papers are about overexpression, and the disorder that you all have encountered is about loss of function, but those are going to be clues. And the way Matthew talked about the Goldilocks effect and the worry about having too much, if you end up with too much gene product, if you have gene therapy, for example, you have to know exactly how much is exactly right. So you've got to get information into Rare X so you can find out if these symptoms are shared in common with all the different families, and if there might even be mutations that cause an excess of FAM177A, which I think we probably don't really know yet if there are those kind of mutations other than in these conditions that show immune issues, maybe some cancer, And there's even a link to, it looks like, to heart disease when there's an overexpression. So all of these things are really interesting to and important to find out. So this is just a quote by Janet Woodcock, who was commissioner of the FDA and instrumental in really trying to turn the FDA around to being more patient-centered, and just showing that harnessing the type of knowledge that you put in natural history studies is really essential for making clinical trials that will actually be approved. So, what's the difference between natural history versus registry? A registry is one point in time, and it's nice. It's good to have a registry. It's good to understand a few things about somebody and what it's like in their life at that one point. But natural history is longitudinal. That's how we understand how things unfold over time. And so hopefully you all will have treatments coming along before you have to collect 20 years of data, but it doesn't matter. You need to collect repeated measures over time to be able to understand how things change. This can be things like, when do the kids learn to crawl or walk or speak or write or point, or any different thing that they might learn to do? That helps you figure out when you have to treat. If there's a window where you absolutely have to treat before that to be able to reverse some of these symptoms. So you've got to collect natural history longitudinally. And you can do that on the RareX platform. I'm going to skip this one. Natural history is basically collecting information about the person with the disorder, and sometimes about the family as well, with or without treatment. Just collecting information and data about their their medication, their treatments, their symptoms, and their therapies. Just collecting it, not intervening, just learning. Natural history, that's what's natural about it. There are a lot of different things you can do in natural history besides plan clinical trials. You can also use that natural history to create standard of care, so that if a doctor has a kid diagnosed with this mutation, they could hopefully go online, look up the standard of care for FAM177A1, and then pull up a whole list of symptoms and the way that they're best treated. You can use it for genotype phenotype correlations, so you can try to understand where the mutation is on the gene, exactly what symptoms it affects. You can also, like I mentioned a minute ago, create a timeline of the disorder, so you know when you have to intervene to prevent which symptoms. And then this is the part that I like the most, because it goes back to the outcome measure and biomarker thing. You can also use natural history to measure change and to measure whether or not a new treatment that you're using is actually working. And you all have a rare disease. This is a nano rare disease. And so with a nano rare disease, you don't have a lot of families around who can serve as placebo controls. Almost every family that you're going to encounter is going to need to be in a trial to be able to have enough numbers to have those trials make sense. And so if you have really good natural history data that you've collected already over time, sometimes in a nano rare disease especially, you can use that natural history data as a control. So this is a slide that Tanya Brown from the Test Foundation made, which is showing how in traditional clinical trials you have to use a placebo group. But in clinical trials with really robust natural history, the whole idea is to do it well enough that you can even use that as a control. And that means that instead of getting worse over time, when a kid gets a treatment, if you can show that the kid is progressing a lot slower than a kid who doesn't have any treatment, even just that could be labeled as success and can get a drug through a clinical trial. And this was done for spinal muscular atrophy, with which Matthew mentioned a minute ago. Um, because spinal muscular atrophy is such a severe disorder, and there are certain parts of certain types of spinal muscular atrophy that are really fatal fast. Because of that, they were able to use the natural history of spinal muscular atrophy as a control. And so we were able, as a community of patients and scientists, not me personally, we were able to get the drug through for SMA much faster because of the amazing natural history that had been collected over about the disease. And this is just a slide that shows you, really, they were looking at how many patients had not needed to go on the ventilator during the time that they were treated with Zolgensma. And so what it shows in the gray is kids that were not treated, and how often they had, or how soon they had to be placed on the ventilator. And in green, this shows the ones who did not, and you can see that it was really, really successful, and that gray line came from natural history. And then this is just an example of one of the, I don't know how many natural history studies I've put my kid in. He's a guinea pig for everything that has anything to do with Angelman syndrome. And this is my kid here in the picture on the right hand side. And one of these little bitty red lines is him. And I'm very proud of that little bitty red line, because that little bitty red line contributes to the natural history of Angelman syndrome that helps us understand where kids with Angelman syndrome are at different ages. And if we give a treatment, and right now there are 3 antisense oligos that are in clinical trials, plus several different drugs and even new drugs. And so if my kid with a little bitty red line is able to move up into this section with the blue lines, that will be a huge success. The section with the blue lines are the milder phenotypes. The section with the red lines are the deletions, which have a more severe phenotype. Even if he could just move into the section which is the milder phenotype, it would be life-changing for us. And we can show this because of natural history. So I'm almost done. I was trying to speed through. Natural history is a hassle. I personally hate doing it. I have to do it all the time. I have 3 surveys waiting to fill out today. It's annoying. It's aggravating. And you know what? It is so worth it. There is no immediate benefit, but it is the most powerful thing the families own. Knowledge about themselves, knowledge about their own kids. It is when they can change science, they can bring treatments to their own kids. So the power is in your fingertips. And here's the link right here, which I pulled from your website, and it leads you to the Rare X link, which leads you to the FAM177A1 portal, and you can get started right away. It's a lot of surveys. It is a hassle. Just do it. I have another thing to tell you, and that is there's power in your blood. So a minute ago or an hour ago, Ethan was talking about biomarker development. Often, to get really nice biomarkers, and the way that David Fagenbaum did it with SomaScan is through plasma. To get plasma and to process it properly for a biomarker, you need to spin it down and flash freeze it on dry ice within an hour of collection. That can be sort of complicated. So at Combined Brain, we have a new way of doing this. And any family with your disorder or any disorder that's in Combined Brain can walk into any of these other family conferences for other disorders and do a donation of a biosample. They can give plasma, you can order an extra tube that you can then process into PBMCs. We can collect whole blood, we can collect dried blood spots, and we can collect urine. And all of those can be put in the biorepository. Your foundation owns them, and they may be included in the biomarker project if you want them included in the biomarker project. And that data will then be returned back to the foundations, and the foundation can then send it back to all your scientists or whoever wants it to use that to develop the next phase of drug development. So that is where I'm going to stop. And I really appreciate everyone listening after the end of when I was supposed to be talking. And thank you very much.
Thank you, Terry Jo. I told you she would not disappoint. You're so inspiring. And I'm reminded that one of the disorders we ruled out for Charlotte was Angelman's. And I remember at the time hearing that the Angelman's community had a mouse. And I was thinking, what? Like, it was so science fiction to me. It was so crazy. And now here we are. It's really amazing. And we're going really fast. And the Angelman's crew is they're best in class. So we're following what you're doing, and we're thankful for the work you're doing.